All right, today we're going to unpack a really fascinating medical discovery from a major clinical trial. It's something that could totally change how we fight one of the toughest forms of breast cancer, all by using a simple test to find a hidden biological clue.
We often think of a cancer diagnosis leading to a pretty standard treatment plan. But the reality is, the same medicine can work wonders for one person and do almost nothing for another. The big question for clinicians has always been, how do you figure out, ahead of time, who is going to benefit and who isn't?
So our story really starts with this particularly aggressive form of the disease called triple-negative breast cancer, or as you know it, TNBC.
So what's the deal with TNBC? What makes it so tough to treat? Well, most breast cancers have these specific molecular locks on their surface, receptors for estrogen, progesterone, or a protein called HER2. And all our modern targeted drugs are designed like keys to fit those locks. But TNBC, just like the name says, is negative for all three.
And this right here is the core of the problem for us as doctors. For most breast cancers, you have clear targets to aim for. But with TNBC, nope. Those standard, highly effective options are just off the table. It's kind of like trying to unlock a door that doesn't even have a keyhole.
This leaves us with a really high-stakes question in the clinic. Chemotherapy is our main weapon here, but there's a specific drug, capecitabine, that's a real puzzle. For some patients, it significantly improves outcomes, but for others, it just adds toxicity with no benefit. So the question is simple but crucial. Who should get it?
To solve this puzzle, researchers started digging for a clue. They had a hunch that TNBC wasn't really just one disease, but probably a bunch of different subtypes all hiding under the same name. They just needed a practical way to tell them apart.
Their main hypothesis was that the secret was right there, in these hidden subtypes. They suspected TNBC could be split into two main groups, basal-like, or BLBC, and non-basal-like. And they really believed that being able to tell these two apart was the key to predicting who would actually get a real benefit from capecitabine.
The problem, though? The gold standard test for identifying these subtypes, PAM50, is a pretty complex genetic analysis. It's expensive, it's slow, and it's just not available in most hospitals. So, researchers came up with a really clever idea. What if a simple, widely used lab test, just a standard immunohistochemistry, or IHC, could do the same job just by looking for a single protein called FOXC1?
So to test this clever and frankly very practical idea, they turned to the data from a major international study. This was the GICOM-CIOBIOMA clinical trial, which had already tested capecitabine in exactly this patient population.
The setup here was absolutely perfect for answering their question. They had these archived tumor samples from over 700 women in the original trial. Half of them got the extra capecitabine, and half didn't. They then used this new, simple FOX1 test to sort every single tumor into one of two buckets, low FOX1 or high FOX1. The goal was to see if that simple grouping could predict who benefited from the drug. And this is where it all comes together. After all that setup, the results from this incredibly simple test gave a really powerful answer to that very complex clinical question.
So the main question was crystal clear. Could this straightforward, affordable protein test really tell us which patients would get a real benefit from an extra round of chemotherapy?
And the answer came in one powerful number. For patients whose tumors had low levels of that FOXC1 protein, the hazard ratio for distant relapse was just 0.44. Now, in clinical trials, a hazard ratio below 1 means reduced risk. A number like 0.44, well, that's a very, very strong signal.
So what does a number like that actually mean for a patient? It means that the patients identified by this test as having low FOXC1 tumors, who then got the capecitabine, saw their risk of a distant, life-threatening relapse cut by 56%. That is a massive, clinically meaningful benefit.
And what's really crucial here is that this wasn't a fluke. The researchers looked at the data from every angle, from distant relapse-free survival to overall survival, and the benefit for this low FOXC1 group was strong and consistent across the board. The test was reliably picking out the right group of patients.
So let's talk about the clinical bottom line. How does a discovery like this go from being a research paper to something you can actually use in your practice?
Well, this test is extremely promising. It's cheap, it's accessible, and it appears to be a powerful predictor. But, good science is always cautious. The researchers are very clear that these findings need to be confirmed in future trials. And they also point out some limitations of the original study, like having only a small number of patients with stage 1 disease.
And that brings us to the key takeaway. For the first time, this study gives us strong evidence that a simple, affordable test could become a practical tool for personalizing treatment. We could potentially use it to confidently identify which of our patients with triple-negative breast cancer are most likely to benefit from adding capecitabine to their therapy.
And that really leaves you with a final, powerful thought. This discovery isn't just about one drug or one type of cancer. It's about a future where we can look for these hidden biological clues, these simple protein signatures, to unlock the unique biology of every single tumor and fight cancer in a much more intelligent, personalized way.